Photosynthesis. This is the overall equation for photosynthesis. We have 6 carbon dioxide and 6 water, with the help of light turning into sugar and 6 oxygen. These are some plant cells. Underneath the microscope you can see the chloroplasts. Chloroplasts are fairly large in size. If we take a look at an artist's drawing of the chloroplast, we have a couple parts that we need to know. You have the outer membrane here. The inner membrane, similar to that of the inner membrane of the mitochondria, are the thylakoids. These pancakes are thylakoids. This is where the electron transport chain takes place. A stack of thylakoids is called a granum, and grana is plural. Grana is several granum. The area surrounding the thylakoids or the grana is called the stroma. This is analogous to the matrix of the mitochondria. Within the thylakoid of the chloroplast is chlorophyll. This is a picture of the molecule of chlorophyll. It has these long tails of hydrocarbons that have those hydrogens that contain electrons that get excited when light hits them. And they will be used for the electron transport chain of the light-dependent reactions. We have 2 types of chlorophyll. We have chlorophyll A and chlorophyll B. Chlorophyll A and chlorophyll B utilize different wavelengths of light in order to make a broader range of wavelength of light that they pick up. So why are plants green? If you take a look at the graph here, along the x-axis is the wavelength of light. On the y-axis is how much light is absorbed. The higher the peak, the more light that's absorbed. If you notice, the highest absorption is here in the reds, and then also down here in the blues. Green is not very well absorbed. Green light is reflected out, and because it's reflected out, that's what we see. And that's why plants are green. The first reaction of photosynthesis is called the light-dependent reactions. This takes place within the thylakoid. This is a thylakoid. Out here is the stroma. Within the thylakoid is where our chlorophyll is kept. The chlorophyll isn't kept in what we call photosystems. We have 2 photosystems. We have photosystem 2 and photosystem 1. Photosystem 2 occurs first, photosystem 1 occurs second. Photosystem 1 was discovered first, and photosystem 2 was discovered second. Light comes in and hits photosystem 2. The electrons on the chlorophyll, on those long tails of the chlorophyll, get excited, and they raise in energy, and they're going to bounce down an electron transport chain. Very similar to that of cellular respiration, these electrons are going to bounce down the electron transport chain. A hydrogen gradient builds up. There is a need to kind of go from that high to low concentration of hydrogen ions. They find the ATP synthase and they make ATP. Because we already know the mechanism, we are going to not go over the mechanism again because it does work the same as cellular respiration. We're just going to know that this first electron transport chain uses ATP synthase in order to produce ATP. Now, the electrons that left photosystem 2, that left that chlorophyll, if we didn't replace them, sooner or later they will run out because the chloroplast is not necessarily replaced. So we need to somehow fill in those holes of electrons. We need to replace those electrons. In order to do that, what we do is we take water and we split it. The H's of the water are utilized as the replacement electrons, and they also help with that hydrogen-proton gradient. The oxygen is going to leave as oxygen gas. Now we're on to photosystem 1. Again, light's going to come in and hit photosystem 1. Light is going to excite the electrons on the chlorophyll within photosystem 1. That electron is going to raise in energy. It's going to go down an electron transport chain. This time, it's going to create NADPH. NADPH is an electron carrier similar to that of NADH of cellular respiration. Here, in order to help you remember that NADPH is used in photosynthesis, we can think that the P stands for photosynthesis. It actually stands for phosphate, but in our heads that we can remember that that P is for photosynthesis, that NADPH is in photosynthesis. NADPH is made with the enzyme NADP reductase. The products here, ATP and NADPH, those are important in order to help feed Calvin. Calvin is the next step within photosynthesis. We're going to give Calvin some energy in order to make our energy molecule of sugar. Oxygen gas is a happy byproduct. It's utilized as the final electron acceptor in the electron transport chain. Light-dependent reaction happens within the thylakoid membrane. It utilizes two different electron transport chains. It uses water to replace those lost electrons, making oxygen gas. The first electron transport chain makes ATP, the second one makes NADPH, and those go on to feed Calvin. This is just another picture of the light-dependent reactions. We have our two photosystems, the P680 and the P700. Those are just the wavelengths of light that they utilize, 680 nanometers and 700 nanometers. The light hits photosystem 2. That electron raises up in energy. It bounces down an electron transport chain. During that time, it makes ATP. We have light hitting photosystem 1. That electron raises up in energy, bounces down an electron transport chain to make NADPH. Those two go on to Calvin. This is the Calvin cycle. This is a little bit beyond what you need to know, but there are a couple key points here that we have to go over. Carbon dioxide here is fixed to a molecule called RUBP. RUBP stands for ribulose bisphosphate. You don't need to know that. You just need to know that carbon dioxide gets attached to this other molecule called RUBP. So RUBP and CO2 come in. Rubisco is an enzyme. Rubisco stands for ribulose bisphosphate carboxylase. Again, you don't need to know that, but Rubisco is an enzyme that fixes carbon. That fixes carbon dioxide to RUBP. Then we add in our ATP and our NADPH. We generate G3P, which is our 3-carbon molecule, which when we add 2 G3Ps together, we're going to get our glucose molecule. And then we have to regenerate RUBP, so we need to put in some more ATP. Let me go ahead and redraw this for you to be a little bit more clear. The Calvin cycle in the stroma has carbon dioxide being fixed to RUBP. So carbon dioxide is attached to that RUBP. The enzyme that helps do this is called Rubisco. We are going to put in some energy in the form of ATP and NADPH. This came from the light-dependent reactions in the thylakoid. We make our G3P. G3P is going to go on to make our glucose. So 2G3P equals 1 glucose. We need to regenerate the RUBP, so we need to put in a little bit more ATP, and we'll regenerate that RUBP and start over again by fixing another carbon dioxide with the help of Verbisco. Let's talk about the factors affecting photosynthesis. If you remember the reactants or the things that go into photosynthesis, if we're lacking any of those, we're going to slow down the rate of photosynthesis. If we are lacking water, we're going to slow down photosynthesis. We need that water in order to replace those electrons that get lost from the chlorophyll. Sunlight, the amount of sun that we have, is going to affect the rate. Also, the type of sun rays that the plant is getting can make a difference. If you were to grow your plants under green light only, your plants would not be very successful. Temperature. Generally, in lower temperatures, photosynthesis will slow down. In warmer temperatures, it will speed up until it gets to a certain point, and then it gets too hot for photosynthesis to occur. So H2O levels, sun levels, and temperature are all things that generally affect the rate of photosynthesis. And the bulk of photosynthesis, where most photosynthesis happen on Earth, is actually within the ocean. There is a bunch of phytoplankton that lives within the ocean that photosynthesizes, so more than beyond plants. They actually belong to the kingdom Protista. Most of our photosynthesis actually happens within the ocean. Thank you for watching, and we'll see you next time.